In this short video lecture, we can discuss about translation. Translation is a part of protein synthesis which is happening in living organisms. Translation is carried out by ribosomes. Protein synthesis is a complex process having two phases, transcription and translation. During transcription, a messenger RNA is transcribed from DNA, from the coding strand of DNA, and this messenger RNA will undergo splicing and processing to form a functional mRNA, and this mRNA moves to cytoplasm. Cytoplasm is the site where protein synthesis is happening. Ribosome are the organelles which are doing protein synthesis. Now let us see in detail what is translation and what are the steps and what are the process happening during translation. Translation is the process by which the base sequence of mRNA is interpreted into amino acid sequence of a polypeptide chain. We know the genetic message on DNA is transcribed to messenger RNA, and the messenger RNA has undergone processing, splicing and processing to remove all the introns. A processed messenger RNA will be carrying genetic message in the form of codons or codes. So the translation is the process by which this message stored in codes or this message which is stored in codons are translated or converted to amino acid sequence and then to a protein. Translation has 4 steps. First one is activation of amino acids. Then the attachment of activated amino acid with transfer RNA or tRNA. Elongation of the polypeptide chain. Termination of the polypeptide chain. Let's see the overview in a nutshell first and familiarize with what is translation. Classically speaking, mRNA attaches to the ribosome. tRNA attached to amino acid. tRNA carries amino acid to ribosome. tRNA delivers the amino acid to ribosome by looking complementarity between codon of mRNA and anticodon of tRNA. If correct, amino acids are released to ribosome. Amino acids are hooked with peptide bond enzymatically. Like this, the protein chain will grow. Messenger RNA genetic message store ചെയ്യും. ആ genetic message അനുസരിച്ച് tRNA കൊണ്ടുവരുന്ന അമിനോ ആസിഡുകളെ ചേർത്ത് പ്രോട്ടീൻ ഉണ്ടാക്കും. ആദ്യമായി മെസ്സഞ്ചർ RNA റൈബോസോവുമായിട്ട് അറ്റാച്ച് ചെയ്യുന്നു. ട്രാൻസ്ഫർ RNA അമിനോ ആസിഡുകളെ റൈബോസോമിന്റെ അടുത്തോട്ട് കൊണ്ടുവരുന്നു. കോഡോണും ആന്റികോഡോണും മാച്ച് ചെയ്യുകയാണെങ്കിൽ അമിനോ ആസിഡുകളെ റൈബോസോമിന് കൊടുക്കുന്നു. ഈ അമിനോ ആസിഡുകളെ ചേർക്കുന്നത് പെപ്റ്റൈഡ് ബോണ്ട് കൊണ്ടാണ്, അത് എൻസൈം ഉപയോഗിച്ചിട്ടാണ്. അങ്ങനെ ഒരു പ്രോട്ടീൻ ചെയിൻ കംപ്ലീറ്റ് ചെയ്യുമ്പോൾ അത് റൈബോസോമിൽ നിന്ന് റിലീസ് ചെയ്യുന്നു. We can see in detail how this process is happening. Translation, as we all know, is happening in റൈബോസോം. റൈബോസോമിലാണ് ഇത് സംഭവിക്കുന്നത്. And the final product of translation is a polypeptide. We know during this process, ribosome moves from 3' to 5' in the mRNA. mRNA പുറത്തൂടെ റൈബോസോം 3' 5' മൂവ് ചെയ്യും. അപ്പോൾ അത് എപ്പോൾ മൂവ് ചെയ്താലും 3 കോഡോണ് കവർ ചെയ്താണ് മൂവ് ചെയ്യുന്നത്. വൈ? ബിക്കോസ് കോഡോൺസ് കണ്ടെയ്ൻ 3 ന്യൂക്ലിയോടൈഡ്സ്. സോ വെൻ എവർ റൈബോസോം മൂവ്സ്, ഇറ്റ് മൂവ്സ് 3 ന്യൂക്ലിയോടൈഡ് അറ്റ് എ ടൈം ടു ഹാവ് ദി സിഗ്നൽ. ആ 3 ലെറ്റർ കോഡാണ് ഒരു അമിനോ ആസിഡ്. അതുകൊണ്ട് എപ്പോഴും 3 ലെറ്റർ കോഡിലോട്ടായിരിക്കും മൂവ് ചെയ്യുന്നത്. Now let's see in detail what are the steps of translation. As I told you before, there are 4 steps. First one, activation of amino acid. We are familiar that amino acids are the building block of proteins. But whatever reactions, these are all biochemical reactions, and whatever reactions happening in living system needs energy. So amino acids are activated with ATP, or amino acids are energized with ATP before they are transported by tRNA. tRNA amino acid ne കൊണ്ടുവരുന്നതിന് മുമ്പ് amino acid ne ATP ഉപയോഗിച്ച് energize ചെയ്തു. And this activation of amino acid with ATP is called amino acylation. The activation of amino acid with ATP is called amino acylation. And it is carried out by the enzyme amino acyl tRNA synthase. Amino acyl tRNA synthase. So, in activation of amino acid, an amino acid combines with ATP in the presence of amino acyl tRNA synthase to form amino acyl tRNA. So, activation of amino acid gives amino acyl, or activation of amino acid gives charged amino acid which can combine with tRNA. The second stage is attachment of activated amino acid with tRNA. Transfer RNA or tRNA are the agents which carry corresponding amino acid from cellular pool to ribosome. Protein synthesis നടക്കുന്നത് റൈബോസോമിലാണ്. സെല്ലുലാർ പൂളിലാണ് അമിനോ ആസിഡ് കാണുന്നത്. ഈ സെല്ലിലുള്ള അമിനോ ആസിഡ് പൂളിൽ നിന്ന് അമിനോ ആസിഡിനെ കറക്റ്റായിട്ട് ക്യാരി ചെയ്യേണ്ടത് tRNA മോളിക്യൂൾസ് ആണ്. അപ്പം അങ്ങനെയാണെങ്കിൽ tRNA മോളിക്യൂളുമായിട്ട് അമിനോ ആസിഡ് ചേരുന്നതും ഒരു ബോണ്ടിങ് ആണ്. അതിനുവേണ്ടിയുള്ള എനർജിയാണ് നമ്മൾ അമിനോ ആസിഡിന് കൊടുത്തത് ATP വഴി. സോ നൗ ദി ആക്ടിവേറ്റഡ് അമിനോ ആസിഡ് അറ്റാച്ച്ഡ് ടു ദി amino acid acceptor arm of tRNA. Each amino acid very specific. A particular tRNA can bind to only a particular amino acid. So amino acids are specific to tRNA. Like exactly tRNA is also specific to amino acid. So only a specific amino acid can bind to its own tRNA. So once the specificity is there, an activated amino acid binds to acceptor arm of tRNA. And this is the acceptor arm. The acceptor arm of amino acid binds amino acid to it. Now, and when an activated amino acid binds to tRNA, it is called amino acylation of tRNA. Amino acylation. This process of fusion of amino acid to tRNA is called amino acylation of tRNA. And this reaction or this fusion of amino acid to tRNA is catalyzed by the enzyme amino acyl tRNA synthetase. Amino acyl tRNA synthetase. So now an amino acyl tRNA complex is formed. By the fusion of activated amino acid with tRNA, an amino acyl tRNA complex is formed. The third step is initiation of polypeptide chain. The initiation of polypeptide chain is by the transfer of first amino acid to ribosome. When the first amino acid reaches ribosome, we can tell polypeptide chain synthesis started, and that is the initiation of polypeptide chain. For this process, the machinery for protein synthesis is ribosome subunit. So we need ribosomes. tRNA has to bring amino acids, so there should be amino acyl tRNA complexes. The message is carried by mRNA, so there should be mRNA. And also, for the initiation of polypeptide chain, lot of initiation factors are required. As the first stage, ribosomes are having two subunits, 30S and 50S in prokaryotes. The 30S ribosome binds with initiation factor 3, forming a complex 30S IF3 complex. This is the first step of initiation. 30S ribosomes bind with the initiation factor 3, forming 30S IF3 complex. So these are the two subunits. Smaller unit is the 30S unit. At first, a factor, IF3 factor, will come and bind with the ribosome, forming 30S IF3 complex. Once 30S IF3 complex is formed, it binds to messenger RNA to form 30S IF3 messenger RNA complex. So 30S unit is the smaller subunit of ribosome. With the factor, with an initiation factor, it gets combined with mRNA to form 30S initiation factor mRNA complex. Whenever ribosome binds with messenger RNA, always the first codon, the initiation codon is AUG, that stands for methionine. Initiation codon is AUG and it stands for methionine. So whenever an mRNA binds with ribosomal complex or smaller subunit of ribosome, within the P site of ribosome is having P site, E site and A site. There are three sites are present in ribosome: P site, A site and E site. P site is called peptidyl site. When the first mRNA or when the ribosome first attached to mRNA, it is attached to the P site of ribosome and the ribosome is fixed. Ribosome will be fixed in a position where the first codon AUG is present. Whenever ribosome comes and fixed to mRNA, or see, this is the messenger RNA and this is the ribosomal units. See, this is the smaller subunit. The ribosome always aligns and fixes on the position of AUG, that is the initiation codon. Now, once smaller subunit of ribosome aligns with the codon AUG through the P site of ribosome, from the cellular pool, the tRNA which carry methionine or methionine tRNA binds to initiation factor 2 and GTP, forming tRNA complex. So there is a tRNA which can carry methionine. This tRNA binds with methionine in the presence of initiation factor 2 and GTP. Initiation factor 2 GTP ഉപയോഗിച്ച് methionine അതിന്റെ corresponding tRNA ആയിട്ട് bind ചെയ്യുന്നു. Now this messenger RNA complex. What is messenger RNA complex? It's formed of 30S ribosome, IF3 and messenger RNA. That complex binds with tRNA complex and that forms pre-initiation complex. So pre-initiation complex in protein synthesis is the binding of mRNA complex with tRNA complex. So mRNA complex binds with tRNA complex to form a pre-initiation complex. So the mRNA complex binds with tRNA complex, forming pre-initiation complex. So just classically speaking, mRNA forms a complex, tRNA forms a complex. mRNA forms a complex with smaller subunit with one initiation factor. tRNA forms a complex with amino acid and that needs energy, so GTP is there and another initiation factor is also associated. Now, once the pre-initiation complex is formed, the larger subunit of ribosome come and fuse to the smaller subunit to form a complex. Ribosome get functionalized only when the larger subunit and smaller subunit are combined together. When there is pre-initiation complex formation, only smaller subunit is there. But once the pre-initiation complex is formed, the larger subunit comes and binds to the smaller subunit and the pre-initiation complex to form the functional ribosome. A functional ribosome has three sites. One is called A site, second one is called P site, and the third one is called E site. A functional ribosome has three sites. One is A site, P site, and E site. A site is called amino acyl site. P site is called peptidyl site. E site is called exit site. A site is also called acceptor site because it accepts tRNA complex. Whenever tRNA carries an amino acid and comes to the ribosome, it is received by the amino acid site. P site is called a polymerization site. The polymerization, that is peptide bond formation, happens in P site. So peptide bonds are the bonds which combine amino acid together to form a peptide bond. So that is the polymerization site. P site polymerizes peptide bond formation. Third one is called E site. Once tRNA brings amino acid, and after peptide formation, after giving the amino acid to ribosome, tRNA will go away. And this tRNA is released. The unloaded tRNA is expulsed or released through this E site or exit site. So A site for amino acid acceptance, P site for peptide bond formation, E site for exit. But there is a difference. At the beginning of protein synthesis, the first amino acid, first tRNA is received through P site. Only the first tRNA is accepted to P site. All other tRNAs are accepted through A site. So once the complex, the ribosomal complex and the tRNA complex combines, and once the larger subunit unite with the smaller subunit of ribosome, the chain elongation, the ribosome is ready for doing the chain elongation. What is elongation? Elongation refers to the addition of amino acid one by one sequentially, and then a peptide bond will be formed sequentially. So, but the first amino acid will be methionine. mRNA first amino acid methionine codon ആയിരിക്കും. അതുകൊണ്ട് first amino acid methionine ആയിരിക്കും. Once the first amino acid is accepted by the ribosome moves forward. Ribosome ഒന്ന് മുന്നോട്ട് നീങ്ങും. അടുത്ത കോഡോൺ, അതായത് റൈബോസോം 3' 5' ഡയറക്റ്റിൽ നീങ്ങും. അപ്പോൾ ആദ്യത്തെ കോഡോൺ റൈബോസോം റീഡ് ചെയ്തു, അത് മെത്തിയോണൈൻ ആയിരുന്നു, അതിൻ്റെ അമിനോ ആസിഡ് അതിന് കിട്ടി. നൗ ഇറ്റ് മൂവ്സ് ടു ദി സെക്കൻഡ് കോഡോൺ, അടുത്ത 3 ലെറ്ററിലോട്ട് പോകും. അപ്പോൾ അവിടെ റെക്കഗ്നൈസ് ചെയ്യും അത് ഏതാണ് കോഡോൺ എന്ന് അമിനോ റൈബോസോം റെക്കഗ്നൈസ് ചെയ്യും. Suddenly the corresponding tRNA brings the amino acid from the cellular pool and it enters to the amino acid acceptor site or A site. And if it fits, if the codon and anticodon complementarity is okay, and if it fits, the tRNA complex is accepted by the A site, and the amino acid is released to P site, and on the P site a peptide bond is formed, and this bond formation is carried out by the enzyme peptidyl transferase, and this bond formation is carried by the enzyme peptidyl transferase. After formation of this peptide bond, after giving this amino acid to the ribosome, the tRNA is released by the enzyme deacylase. So tRNA carries amino acid, and once it gives amino acid to the P site and peptide bond is formed, the amino acid is released by the enzyme. The amino acid is released from tRNA, or tRNA is released from amino acid by the enzyme tRNA deacylase. Now the tRNA is dissociated or released, and it moves from P site to A site. So the charged tRNA, or the tRNA which carries amino acid, is accepted by A site. It moves through the A site as a complex. At the P site, the amino acid forms a bond with the previous amino acid in the peptide site. Once it is over, deacylase will release the tRNA. It moves through E site to cytoplasm. So the ribosome moves in 3', 5' direction. 3', 5' direction ribosome move ചെയ്തുകൊണ്ടിരിക്കും. ഓരോ തവണയും ഒരു കോഡോൺ ഡിസ്റ്റൻസ് നീങ്ങുള്ളൂ. ഈ മൂവ്മെന്റിനെ സഹായിക്കുന്നത് by the enzyme translocase. The movement of ribosome from one codon to another is catalyzed by the enzyme translocase. So this is what is happening. This is the ribosome. First it will bind to AUG. Now once the first amino acid added, it moves further, it moves forward. When it moves forward, when it moves in 3', 5' direction move ചെയ്യുമ്പോൾ corresponding amino acid വരുന്നു. All these amino acids are linked together to form a polypeptide. As the ribosome moves on the messenger RNA and it reads the codon, it recognizes the codons, the corresponding amino acyl tRNA supply the amino acid, and the peptide bond formation will continue. During this process, and this was the first ribosome. ഒരു ribosome attach ചെയ്ത് മൂവ് ചെയ്ത് മുമ്പോട്ട് പോകുമ്പോൾ അടുത്ത റൈബോസോം അതിൻ്റെ പിറകെ വന്ന് അറ്റാച്ച് ചെയ്യാം. അങ്ങനെ ഒരു മെസ്സഞ്ചർ ആർഎൻഎയിൽ മൾട്ടിപ്പിൾ റൈബോസോംസ് വന്ന് one after another ആയിട്ട് വന്ന് മൾട്ടിപ്പിൾ റൈബോസോം ഒരു mRNAയിൽ അറ്റാച്ച് ചെയ്ത് നിന്ന് പ്രോട്ടീൻ സിന്തസിസ് നടത്താറുണ്ട്. ദിസ് കണ്ടീഷൻ in which multiple ribosomes are attached to an mRNA is called polyribosome. Polyribosome or polysome. See, what is this recognition? This messenger RNA is having 3-letter code, and that is called codons. tRNA has 3-letter codons on the top, that is called anticodons. Only if this codon and anticodon matches, then only the corresponding amino acid accepted. Otherwise, if it is not correct, the amino acid will not be accepted by the ribosome. The specificity of this reaction, protein synthesis, is codon anticodon recognition. The process will continue, and finally the chain has to terminate. Chain terminate ചെയ്താലേ ഈ പ്രോസസ് കംപ്ലീറ്റ് ചെയ്യുള്ളൂ, പെപ്റ്റൈഡ് സൈറ്റോപ്ലാസത്തിലോട്ട് പോവുള്ളൂ. So termination is the completion of synthesis of polypeptide chain. Termination എന്ന് പറഞ്ഞാൽ polypeptide chainന്റെ termination completion ആണ്. Once the ribosome moves and reach the stop codon, അതായത് റൈബോസോം ഇങ്ങനെ മൂവ് ചെയ്ത് റൈബോസോം മെസഞ്ചർ ആർഎൻഎയിലൂടെ നീങ്ങി അവസാനം അത് സ്റ്റോപ്പ് കോഡോണിലെത്തും. ഏതാണ് സ്റ്റോപ്പ് കോഡോൺ? UAA, UAG, UGA, ഇങ്ങനെ 3 സ്റ്റോപ്പ് കോഡോൺസ് ഉണ്ട്. അവിടെ എത്തുമ്പോൾ ദ റിലീസിംഗ് ഫാക്ടേഴ്സ് RF1, RF2, RF3 ഇത് 3ഉം സൈറ്റോപ്ലാസത്തിലുണ്ട്. Once the ribosome reaches the stop codon, the release factors comes and help the termination. During termination, the polypeptide will be released to the cytoplasm. The dissociation of ribosome will happen. mRNA is released. Everything after the peptide is formed, the whole ribosome assembly will be dismantled. But mRNA will be there because next or after this released ribosome from the back, next പിന്നെ ഉള്ള റൈബോസോംസും പ്രോട്ടീൻ സിന്തസിസ് നടത്തിക്കൊണ്ടിരിക്കുകയാണ്. അതുകൊണ്ട് mRNA അവിടെ കാണും, കുറെ കഴിഞ്ഞ് mRNA നശിക്കുകയുള്ളൂ. So from a single mRNA, multiple ribosomes can synthesize protein. So to brief, you see the steps of translation. An amino acid comes, charged amino acid comes to ribosome subunit, smaller subunit. It aligns. The 2 complexes combine together. This is the initiation complex. and the alignment is in the methionine region. First AUG or methionine region, alignment happens on the methionine region. Once it is aligned and once the initiation complex is formed, the larger subunit comes and binds. And it has different sites: acceptor site, peptide site, and exit site. As it moves further, the second amino acid will come. Next to methionine, valine came here. The process will continue. There will be bond formation. This is the peptide bond. Once the amino acid is released to the ribosome, the tRNA detaches and moves out through the exit site. The ribosome moved further. The third amino acid came there. So it is accepted. Now 3 peptides, 3 amino acids are fused to form a dipeptide. The process continues and it reached the stop codon. Now the chain is released and the amino acid and the ribosomes dissociated. And this is the essence of translation. Thank you very much.